What if a future LDL treatment did not mean a daily pill, a monthly injection, or even a twice-yearly clinic visit? A new early trial is testing VERVE-102, a one-time intravenous infusion designed to reduce PCSK9 production in the liver. The idea is exciting, but the honest version matters. This is investigational, it is early, and it is not an approved treatment. The reason it could matter starts with how your liver clears LDL. This story is about cholesterol biology and gene editing risk, not just a new product headline. VERVE-102 is a base editing medicine from Verve and Lilly. In Heart-2, researchers gave it as a single IV infusion over about 4 hours, not as a routine cholesterol shot. The therapy carries editing instructions into liver cells using lipid nanoparticles. The editor itself is not meant to keep circulating forever. The intended effect comes from the DNA change left behind. The intended edit targets PCSK9, a gene that controls how much PCSK9 protein the liver makes. If that change is durable and safe, the practical question becomes whether LDL can stay lower without repeat dosing. PCSK9 is not a random target. Human genetics made it famous. Some people are born with loss-of-function variants that make PCSK9 less active. Those people tend to have lower LDL cholesterol, and studies found fewer coronary events. The opposite kind of PCSK9 change, gain of function, can push LDL higher and cause familial hypercholesterolemia, which strengthens the target logic. That does not automatically prove a medicine can copy the same benefit, but it gave researchers a strong clue. If you can lower PCSK9 safely, you may preserve more of the liver's LDL cleanup capacity. LDL particles carry cholesterol through the bloodstream. Your liver clears much of that LDL with LDL receptors on liver cells. A simple way to picture them is reusable hooks. A receptor catches an LDL particle, pulls it inside the cell, releases the cholesterol cargo, and can return to the surface. This is why receptor number matters as much as how much cholesterol arrives in the bloodstream. When more receptors keep recycling, the liver has more opportunities to remove LDL from the blood. PCSK9 changes the fate of those receptors. When PCSK9 binds an LDL receptor, that receptor is more likely to be sent to breakdown instead of recycling back to the surface. The breakdown step happens inside the cell, often described as lysosomal degradation, so the receptor cannot make another trip. So, PCSK9 is not destroying LDL directly. It is reducing the number of reusable receptors available to clear LDL. That is why blocking PCSK9, lowering PCSK9 production, or editing PCSK9 at the source can all point toward the same outcome: more receptors and lower LDL. The pathway already has approved drugs, which is important context. Monoclonal antibodies such as evolocumab bind PCSK9 in the blood, so it cannot drag LDL receptors toward degradation. Inclisiran works upstream with small interfering RNA, reducing PCSK9 production in the liver. These medicines can lower LDL substantially, but they are ongoing therapies. Injections every few weeks or monthly for antibodies, and repeated clinic dosing for inclisiran. That prior experience de-risks the target, but not the delivery method, the editing step, or the long-term safety profile of a one-time therapy. VERVE-102 is different because it asks whether one edit could make the pathway stay turned down longer. Now the data. The interim Heart-2 analysis included 35 adults with heterozygous familial hypercholesterolemia or premature coronary artery disease who still needed additional LDL lowering. Participants received one IV infusion across 6 dose cohorts, from 0.3 to 1.0 milligrams per kilogram. Lilly reported dose-dependent mean PCSK9 reductions from 51% at the lowest dose to 88% at the highest dose. Mean LDL-C reductions ranged from 9% to 62%. The highest dose corresponded to an average LDL-C drop of 78 milligrams per deciliter. The study was designed for early safety and biological signal, not to decide whether fewer cardiovascular events occur years later. Those are large early numbers, but this was not an outcomes trial. The delivery system is why this belongs in a different category from a standard drug. VERVE-102 packages messenger RNA for an adenine base editor plus a guide RNA targeting PCSK9. Both are carried in a lipid nanoparticle designed to reach liver cells. The editor is intended to change one DNA letter in the PCSK9 gene, reducing the liver's ability to make PCSK9 protein. In base editing terms, this is a precise chemical letter change rather than cutting both DNA strands. That is also why the bar for safety is high. An intended durable edit needs durable evidence. The caveats are the real story, not a footnote. Phase 1b studies are small and mainly ask, can this be given? What happens to the biology? And what safety signals appear early? Lilly reported no treatment-related serious adverse events or dose-limiting toxicities in this interim analysis, while treatment-related events included low-grade infusion reactions and fatigue. The NEJM report also discusses liver enzyme monitoring. Because the liver is the target organ, liver labs and longer monitoring are not optional details. What we do not have yet is broad experience, years of follow-up, or proof that heart attacks and strokes go down. This is also not a general wellness shortcut for anyone with one borderline LDL result. Heart-2 studied high-risk adults, people with heterozygous familial hypercholesterolemia or premature coronary artery disease who needed more LDL lowering despite oral lipid-lowering therapy. Familial hypercholesterolemia is inherited, so LDL exposure can start early and accumulate over decades. For most people, lifestyle, statins, ezetimibe, and approved add-on therapies remain the practical tools. The reason this trial matters is that high-risk patients often need deep, durable LDL lowering, and adherence to chronic therapy is a real-world problem. The one-time idea changes the trade-off. With approved PCSK9 antibodies, the biology is reversible because the drug eventually clears and another dose is needed. With inclisiran, production is reduced for months, but repeat dosing still continues. Editing aims for a more durable switch in liver cells. That could be powerful if it works, but it also changes the risk conversation. That is the trade. Fewer adherence problems if it works, but less simplicity if an unexpected problem appears. You would want confidence not only in LDL lowering, but in off-target effects, liver safety, durability, and reversibility questions. So the short version is this. PCSK9 limits how many LDL receptors the liver can reuse. Less PCSK9 can mean more receptors and more LDL clearance. VERVE-102 is trying to lower PCSK9 at its genetic source with one investigational infusion. The early LDL numbers are meaningful. The unanswered safety, durability, and outcomes questions are just as meaningful.